Our group was given an unknown solution, and our task was to gather data for an absorbance spectra, which would then be used to determine what dye we were working with and the concentration of the dye in the solution.
Our research question explored if comparing absorbance spectra of our unknown solutions could indicate its identity when compared with absorbance spectra of known solutions. Initially, we predicted we could find the identity of the unknown solution based on its maximum absorption peak. We determined our unknown solution was Sautel's dye because the absorbent peak was at a wavelength of 504 nanometers, which was closest to the peak of Sautel's dye at 475 nanometers. Before moving on, there are some terms we need to define. First, absorbance is the capacity of a solution to absorb light at a specific wavelength. A real-life example of this would be plants appearing to be green due to the fact that the chlorophyll absorbs all light except green, leaving the rest to be reflected. Next, regarding our lab, fluorescence is the emission of light by a solution that has absorbed light. Fluorescence has a clinical laboratory application through the use of flow cytometry, used to diagnose disease. Finally, transmitted spectra results from light that passes through a substance. It can be considered the opposite of absorbance. An easy example of this occurring is picturing light passing through a window.
Beer-Lambert's law describes the relationship between the absorbance and concentration of a solution. As modeled in Figure 1, these variables are directly and linearly related. Furthermore, the slope of a Beer-Lambert plot indicates the product of excitation coefficient and the path length. This relationship can be used to identify unknown solutions. Einstein's photon model of light defines light as a massless tiny particle called a photon. The energy of this particle is affected by its wavelength, which determines which region of the light spectrum it falls in. This spectrum is shown in Figure 2. In this experiment, we observed photons with wavelengths in the visible light region, which was about 400 to 700 nanometers. To model emission, Figure 3 demonstrates how photon absorption causes molecules to jump to higher energy states. When they lose the energy and fall back down to the original energy state, a photon is emitted. The emitted photon has a lower energy than the absorbed photon, and it will be either released into the environment, which Figure 3 shows with a green wave, or absorbed by another molecule, which Figure 3 shows as the black dashed horizontal lines. This emittance of a photon is what causes certain materials to fluoresce.
To find the absorbance of the unknown dye solution, we measured the intensity of light from a flashlight that passed through the solution using a spectrometer. The spectrometer was angled head-on with the light source. Water was measured as a blank, then data was collected for the unknown solution. To collect data about fluorescence, we first measured absorbance by the intensity of light from a flashlight traveling through the solution by lining up our spectrometer head-on with the light source. Emission data was collected by measuring the intensity of light at a 90-degree angle to the flashlight. Figure 6 shows observable emission from the fluorescein resulting from shining a light through the solution.
To find the absorbance, we did A equals log I sub 0 over I, and then we calculated all of our absorbances in Excel, and an example calculation is shown below. To find our concentration, we did A equals E times C times L, and then we rearranged that for concentration. Our absorption number was at the peak of our absorption graph in Figure 7, and then we had two data solutions, which will be mentioned later, and then we found our average concentration from those data. Continuing on, to find the concentration of the fluorescent, we use the same equations as we did for the unknown solution. And then I want to mention that the E sub lambda is a specific extinction coefficient that is different for each solution. So for example, in fluorescent, the extinction coefficient is 92,300. So as I mentioned before, our first absorbance data, which is at the top graph right there, we weren't sure because our peak wasn't like all the other known peaks that we were given. So we redid our data, and that graph is on the bottom, but that was a little skewed too because it had negative values. So we took the average of those two graphs, and then you get the middle pink graph, and that peak was at 504 nanometers. And that was closest to our Sautel dye, which that peak was at 475 nanometers. So we just confirmed that our solution was the Sautel dye. Here in Figure 8, we overlaid the absorbance and the emission spectra to see the wavelengths easier. So there's a peak difference here. So the absorbance and the emission peaks do not overlay each other, and that could be because when a photon gets absorbed on a molecule, the molecule will jump to a higher energy state, and then when the molecule eventually comes down to that lower energy state, it'll emit another photon of a lower energy than it was absorbed originally. And that lower energy photon is the emission spectra. That orange graph right there, it has a longer wavelength, which means a lower energy, compared to the absorbance, which has a shorter wavelength and a higher energy. The use of absorbance spectra can be a helpful tool to identify unknown solutions by comparing their absorbance fingerprint peaks with the spectra of known compounds. And then the concentration of a substance can be determined by the Beer's law. First, by measuring the intensity of a transparent, colorless blank solution to calibrate the spectrometer, and then by measuring the intensity of a known sample. This data can be used to model the Beer-Lambert plot, which will determine the concentration of dye molecules in a solution. The extinction coefficient is a constant for different materials, and then the path length is taken from the experimental cuvette design.
The absorption and fluorescence spectra are different because the absorption spectra indicate the energy of light absorbed by the molecule, while fluorescence spectra display the energy of light released by an excited molecule. The emitted photon has less energy than the absorbed photon. This occurs because light energy was converted into other energy, such as vibrational energy. This form of kinetic energy was represented in Figure 3 by the fourth and seventh arrows. The conversion of light energy into other forms of energy shows that energy is conserved. The limitations of this experiment were centered around the tools used for collecting data. If the light source was not perfectly perpendicular to the cuvette, it made it more difficult for the spectrometer to align perfectly with the light source. This increased the difficulty in obtaining consistent and accurate intensity readings. Other limitations, such as the room not being completely dark and the spectrometer not being properly calibrated, occurred. Despite these limitations, results were obtained and analyzed. Further research, such as observing what happens to fluorescence when the wavelength of light shine on it is controlled, should be performed.